All right, I'm going to be talking today about Chronosequence study. Brandon has also been involved in a little bit of this. He's measured different things and had a slightly different set of biofilters. But what we're really studying here are aging biofilters in Australia. And I should mention before I go on that I'm a postdoctoral scholar at Scripps Institution of Oceanography, and I'm working with NISA 11. So I'm going to be talking today, I'll explain what a chronosequence is in a second. I'll be talking today about invertebrates and metals in natural treatment systems. And primarily, the type of system that I'm covering is shown in this picture here. These are biofilters, which go by a lot of other names, rain gardens, biofiltration systems, and so on, as you can see on the page there. And these systems vary a lot in design and in size. On the upper left in this image, you can see a really small curbside cutout biofilter. And on the bottom right, you can see a really large biofilter in Melbourne at Wicks Reserve, which is actually large enough to actually, support some maybe some wildlife related to some of the things that Rich Ambrose was talking about earlier today. So it's been a lot of research on rain gardens, since they were developed in the 1990s, and or biofilters, as I'm going to refer to them today. And there have been some studies that have been done in the field measuring performance. But a lot of data we get comes from laboratory setups, such as the one that you see in this image here. And more often, what happens, especially now in Southern California, biofilters being required for mitigation whenever new development is built. Some, well, not necessarily biofilters, but some sort of form of treatment to mitigate that new impervious surface. They have to do something to actually allow for collection of that stormwater runoff or mitigation of that extra stormwater runoff, either on-site with something like a biofilter, or they do them off-site with a wetland. The problem that we have with this strategy is that oftentimes biofilters are built, and then there's not much monitoring that goes on afterwards, which is one of the things that Richard was talking about earlier in his talk today. So we have a lot of data sometimes from field monitoring, not often, sometimes from laboratory setups like you see here. And in the laboratory, it's possible to mimic aging with your watering regime, or to run these columns for long periods of time. But there are certain effects of aging that happen in the environment that aren't necessarily replicated or haven't yet been replicated in the laboratory to a great degree. One of those things, and actually, you should see on this image on the right, you can see that Brandon should look familiar to you. We're working in a biofilter here in Melbourne. And one of the things that happens in the natural environment, of course, is that a lot of these biofilters are built along streets, which are also lined with trees. And as you can see here, there's a lot of leaf litter falling into the biofilter directly. So you have this large inputs of particulate carbon that could be affecting the system in ways that Rich to earlier, potentially clogging things, providing food sources for detritivorous organisms that eat organic matter. And also, I think one of the reasons organic matter entering a system might be really important is that it provides a really great binding site for a lot of metals. There are a lot of metals that actually bind really well to organic matter, and that can improve their uptake. Now, the state-of-the-art of design in biofilters in Melbourne now is to use a media normally that is composed largely of fine sand with very low amounts of organic matter present. So even if you start with a system that has very low amounts of organic matter initially, it's going to collect it naturally from the environment over time, from stormwater and also from above, from leaf litter entering the system. And then one of the things that Lisa and I are really interested in are the invertebrates, the animals that live in these systems. And one of the reasons we saw a need for this work, and this is a figure from a paper that we published recently in the Journal of Applied Ecology, there just haven't been many studies on what invertebrates or other animals are doing in biofilters. This is looking over time at studies on, we actually did a literature search using the words biofilter, rain garden, biofiltration to try to find all the papers that we could that were studying these types of systems. Then we searched through them to look for anything that even remotely referred to animals. And we only found 5 studies between 2000 and actually since 1995 until 2013 that looked at animals. And some of these were limited to adding vermicompost or even referring to protozoa within the biofilter, and that might be as far as they got as far as including animals. But as this is actually the same author who Richard was referring to earlier, Izemi has about, I think, 3 papers that they did some work in Melbourne where they actually looked at biodiversity in biofilters of invertebrates. And in all 3, they found that compared to, if you look at this figure on the lower right, Compared to nearby urban gardens and green spaces, bioretention basins, also bioswales in another study, had significantly higher above-ground invertebrate diversity. Now, none of these studies so far have looked at below-ground invertebrates, and because the filtration mechanisms are taking place in biofilter primarily below ground, we might expect those organisms to possibly have effects on function in that region of the biofilter. This is another study that was cited earlier in the session. There's a couple different functions that we're interested in, and in that previous slide, we're talking about biodiversity, and that's a serious issue that is getting a lot of attention in places like Oregon, where they're looking at how they're increasing diversity with rain gardens. But also, there's a potential for animals to have effects on function. And one of the most important functions of biofilters is, improving people actually being able to receive that stormwater runoff that's coming in. One of the biggest maintenance issues that occurs over time in biofilters is clogging. And that's been talked about repeatedly throughout the sessions in this online symposium. Certain plants, as you can see in this here, we're looking at hydraulic conductivity over time. And certain plants, like Carex, which is great at taking up nutrients, you will see a decrease in hydraulic conductivity as the biofilter ages and pore spaces get filled with material. But then you take something like a shrub like Melaleuca, and over time the hydraulic conductivity doesn't decrease, and in fact may even improve over time. So certain plants can actually increase infiltration hydraulic conductivity within a biofilter. There are certain animals that can do that as well, and Lisa Levin mentioned this earlier in her talk. Earthworms, this is a really obvious example, although they're not the only invertebrate that can do this. Earthworms can create tunnels in soil. A lot of people have witnessed this firsthand, and that creates a direct pathway for water to enter the soil, and has been shown in natural soils and even in agricultural systems to greatly increase infiltration rates of water. There have been at least two, one written up in a report, and actually someone contacted Dave McCarthy not too long ago, and that Dave sent that along to Stan, where people working in the field in biofilters have actually noticed anecdotally that they find really good water infiltration rates, and then they look into the media and it is full of worms. And so far that's anecdotal. It hasn't necessarily been demonstrated in a biofilter yet, experimentally. But yeah, so there's other ways though also that earthworms and other invertebrates can affect function in biofilters. And I'm on slide 8 now. Sorry, I haven't been helping people follow along like it should be. But in a number of ways, earthworms, by creating these tunnels, they can actually enhance root expansion by creating bigger pore spaces for plants' roots to grow into. That happens because of the physical space created by the tunnel, and also because the tunnels themselves are lined with high concentrations of nutrients, because earthworm castings tend to have higher concentrations of nitrogen and phosphorus than the surrounding soil. Earthworms and other invertebrates also incorporate organic matter into soil, and they enhance nutrient availability and microbial activity in casts and in tunnels. And so all three of these things can potentially enhance the functions, the nutrient uptake into plants and plant growth that we're wanting to achieve in biofilters. On the lower right, I'm citing a really great review in the European Journal of Soil Science about all the functions that earthworms, all the effects that they have, and ecosystem effects they might have in more natural and agricultural ecosystems. Lisa also alluded to this earlier. Earthworms and some other invertebrates can enhance denitrification. This is on slide 9. And that's a really interesting mechanism by which this happens. We've heard a lot about saturated zones in biofilters throughout the symposium, and these are layers of the biofilter that actually are designed to hold water for prolonged periods of time with a carbon source that helps to lower oxygen levels through microbial respiration. And with low oxygen levels, denitrification is enhanced. And that's usually in that lower level of the biofilter. However, earthworms have anoxic guts. Their gut cavity is entirely without oxygen. And as they take in denitrifying microbes from the soils, those microorganisms enter the anoxic environment, and denitrification rates are ramped up. And as you can see on this figure on the right from a paper by Drake and Horn, both nitrous oxide and dinitrogen gas emissions are increased. These are actually emissions coming directly out of two earthworms, from two different species of earthworm. And it's not only within the worm itself, but they also enhance the nitrous oxide and dinitrogen gas emissions of the soils that they excrete. So those are just a few functions. Lisa and I published, I cited it earlier, the paper in Journal of Applied Ecology that gives a more sort of comprehensive look at all the possible functions of biofilters that might be enhanced by invertebrate activity if anyone's interested in looking at it. But I'm really supposed to be talking about a chronosequence study today. And a chronosequence basically is a study where, because you can't necessarily monitor an ecosystem, or you can, but it takes a very long period of time to monitor long-term changes in ecosystems. And so what you can do in certain situations is actually take a group of ecosystems of different ages and treat them as a time series. So we've done that here in Australia, where we have a chronosequence of 35 biofilters along the East Coast in 3 of the major areas, Melbourne, Sydney, and Brisbane in Australia. This is on slide 10. And we sampled those sites that were, in each city we had a fairly even distribution of ages from 1 to 15 years old, or less than 1 to 15 years old. And we sampled those for a suite of about 26 metals and all the soil invertebrates. And this is work is being done primarily with Belinda Hatt at Monash University. She's our main Australian collaborator and helped design this project. On slide 11. Oh, sorry, if you're following the animation, still on slide 10. So what we're expecting is that as these systems age, so there's the idea that initially when you build a biofilter, you are bringing in media from some sort of a supplier, which may be devoid of invertebrates or may be very low in invertebrate abundance and biomass. Once you build the system, though, invertebrates should colonize from the surrounding landscape, and we would expect species richness to increase with rain garden age. There's other factors that might affect that, which we're going to look at as well. We're also interested in metal accumulation, and so we're looking at things like land use within the watershed, within the catchment of that biofilter, how much is impervious surface. As far as invertebrates, we're going to be evaluating distance to the nearest green space, incorporating island or insular biogeography, which Lisa Levin mentioned earlier in her talk, and also looking at things like plant cover and canopy cover and soil carbon and nitrogen content within the biofilter, how these things are affecting both metal accumulation and biofilter community structure, and potentially how the metals and the invertebrates might be interacting. And just want to quickly, this is on slide 11 now, just quickly wanted to say that there was a large group of people that helped to identify the chronosequence sites, and I think we got a really good site selection because of this, because we had people in the city councils of each different city helping us to pick some really good sites and properly aged sites, because they knew exactly when they'd been constructed. And they're still helping now with getting land use data layers for GIS and spatial analysis, and also any maintenance or design issues that have come up in the biofilters over time. So just initially, we're looking at 3 sites. I'm just going to show a slightly simpler data set where we're looking at 3 sites. This is on slide 12, that we sampled in 2 different seasons. That was winter 2013 and autumn 2014. There's that leaf litter in a biofilter again. And we were actually going and collecting soil cores for, you can see some of our sampling devices, that picture on the left. We were actually collecting soil cores, which we were preserving in formaldehyde for invertebrates. And we're also taking samples that were evenly spaced out spatially at both 2 centimeters or less in depth. Your surface layer of soils were collected for metals. And we also sampled for metal concentrations of 10 centimeters in depth. And our objectives were to, for the invertebrates in this first smaller study, to look at whether or not communities differed among rain gardens, and then whether or not that structure within a site changed with season. And just clicking forward to slide 13. These are the 3 sites that we used. This was actually the first sampling event was during the first up-down under course associated with the UCI Water-PIRE program that took place in Australia. These were 3 systems in Melbourne that were very different from each other. One on the upper left is Hereford Road, which discharges to a stream. Then we have a very large on the upper right and old. It's about 15 years old. This is the first system. This is a bioswale called Lynbrook Boulevard. And that was, I think, the first large-scale demonstration project of green streets and green neighborhoods in Melbourne. On the bottom left, we have a very large biofilter called Wicks Reserve, which is in a more, more outside of the city in a somewhat more natural environment. And keep in mind, again, Hereford Road and Wicks Reserve at the time of sampling were maybe about 4 to 4.5 years old. Lynbrook Boulevard was about 15 years old. So these are actually data. I'm showing these first couple slides on a paper that's been in review for a while now. what we see here, though, on in this first figure on slide 14, is that we're looking at a plot. This is a species rarefaction curve, and we have sample size on the x-axis and expected taxonomic groups on the y-axis. And so this is basically looking at the number of organisms we've collected. And if you see one of these slopes that levels off, like the gray one on the bottom, reaches an asymptote or gets close to it, it means we've probably sampled all the organisms within that system. If you see a line that increases and never quite levels off, it means that we've collected, even though we're getting up over 150 organisms, there were still a lot of species out there that we hadn't actually identified, that we'd somehow missed in our sampling. And suggests that the real species richness, or the number of species out there, are higher than we've actually sampled. But what we can see here is that we've collected, as I say at the top of the slide, there were 29 taxonomic groups, and these were identified to as low as family. So there's a lot more diversity potentially hidden in there in 19 different orders. And our oldest site, Lindbergh Boulevard, in blue, also has the highest species richness, and it looks like there are actually a lot of other species hidden out there that we may have still been yet to collect if we would have taken more samples. Hereford Road and Wicks Reserve were starting to level off and had slightly lower species richness. They were younger sites, so it suggests that maybe the idea that richness increases as these biofilters age could hold true. But again, these are only 3 systems that we sampled here. So you know, it's a rather small, somewhat small study to making a conclusion like that. Now, what do we find? If I'm on slide 15 now, there's a plot on the upper left side of the screen that has abundance, and you can see that we have very large abundance of organisms in these biofilters, especially in Wicks Reserve. We have things, you know, well over 25,000 individuals per square meter, and if you can imagine a square meter on the ground, that's a lot of invertebrates in a small surface area. And the main groups that we have present, if you're using animations, you should click through, and you'll see that there are 4 groups of organisms that were found almost everywhere. And this is true for most of the biofilters that we've sampled. But we have from the upper right to the lower left, we have mites, springtails, earthworms, which are one of our ecosystem engineers that Lisa mentioned earlier in the soils, and then potworms, which are sort of like smaller earthworms. They're much, much smaller, normally about 1 centimeter in length, whereas earthworms can be much larger, and sometimes, you know, several inches in length. So the abundance that we see here, we have these potworms, the Enchytraeids in black, being the most abundant. But if you were to look to put this in terms of biomass, the earthworms are outnumbering everything, or have the greatest biomass by far in these systems. There's not a huge change in the structure between seasons, as you can see on that graph in the upper left. Okay, on slide 16, what you can see here is a multi-dimensional scaling plot. And if you click to get the first animation, I've just drawn circles around points that are from the same sites. And what this does is, these are basically points in 2-dimensional space that are representing the entire community structure. Each point represents the community structure of one site. And the closer 2 points are together, the more similar the community structure or the types and numbers of organisms in those 2 sites are. So we have Lindbergh Boulevard in the blue on the left, and for all these, the filled symbols are in winter, and the hollow symbols are in autumn. In blue, we have Lindbergh Boulevard. In black on the upper right, we have Hereford Road, and in gray on the lower right, we have Wicks Reserve. And what you can see is that these points cluster together fairly well, so the sites have some similarity within samples collected from the same site. But for the most part, except for 2 points that seem to be sort of switched between sites, they have a structure that is different, or that is actually significantly different. We use other statistics to test this. They have a community structure that's significantly different from the other biofilter sites. So there's a structure that seems to be different and also somewhat stable over short time periods between different biofilters. So that was slide 16. Now moving to the next one, slide 17. Those are some of the results. I don't want to go into more detail about that study from some of the some of those early samples we took from those 3 different systems. But now we finally just recently finished going through all the cores from the entire Chronosequence, and that's been completed. And now Lisa and I are starting to dig through there and see if we can get the taxonomic resolution or the identification of the organisms down to as low a level as possible. And we're doing some of the more in-depth analyses that we're looking into with the Chronosequence. And I have to thank on the lower right on this slide 17. Well, there were 2 undergraduate students, Diana Kai-Kittikun and Barbara Aurelio, who did a large number of samples. who helped with this sorting, and Jen Gonzalez has done a massive amount of sorting of these soil pores. And Lisa doesn't cut any corners with sorting. I've come from a stream research background where we split samples and identify things from small fractions to try to save time, but you can miss things. And so here in this lab, we actually go through everything. So I'm very confident that every single organism that was in these samples has ended up in a vial is being counted. And it's a time-consuming process, but you get some great data as a result. So our hypotheses about within this chronosequence is that we think that invertebrate richness, and if you're using the animations, you should click and that'll appear where Jen and Barbara were. If you're using we're thinking that invertebrate richness and abundance should be positively correlated with biofilter age and carbon content. The reason we're assuming carbon content is important is because most of these organisms feed on dead organic matter, and so that inputs of organic matter, carbon in other words, are going to be a source of food. And with age, things have more time to colonize, as suggested by the results we saw from that smaller study that I just presented some results from. And then we think that it is possible that invertebrate richness and abundance will be negatively correlated with metal concentrations in the filter media. So looking at some of those results, if you don't have the full PowerPoint, you're probably seeing everything at once, but right now you should be seeing just a figure in the upper left-hand corner, and that's looking at biofilter age in years on the x-axis with taxonomic richness, and that's just in orders. So there's a lot more diversity probably encapsulated in there on the y-axis. And well, we hypothesized that taxonomic richness would increase with age, but that does not, well, you can use your imagination that maybe there is some sort of an increase there. I forgot to include a p-value, but that's not a significant increase, in case you're wondering. So really, what we're seeing instead, which is potentially more interesting, is that even in systems that are 1 year old, and some of these were actually slightly less than 1 year old, you're actually seeing fairly high taxonomic richness. You're seeing a lot of organisms there present right off the bat. So if they are having effects in biofilters, if invertebrates are having effects in biofilters, they're having them very early after construction, potentially. That is, if they're having substantial effects, which is an important qualifier. Oh, and before I go on, I should mention, I mean, one of the potential reasons for them being there, there's two possibilities for them being present so early on. Is one, they colonize very rapidly from the surrounding environment, which could certainly be the case if they're close to some other green space like a park. Or it's possible, and this is true all over the world, that a lot of organisms are transported with plants, with potted plants, and actually that's one of the ways invasive species make it around the world. So there could be organisms coming in on day one when the plants are actually put into the biofilter, and you could have a fairly large community coming in very early on just by being moved physically by humans. If you click through the animations on the lower left, there's a plot that shows filter media content and taxonomic richness on the y-axis again. Filter media content in milligrams per kilogram is on the x-axis. And there is a significant logarithmic relationship. As filter media content increases, our taxonomic richness increases significantly. Click through again on the y-axis. I'm going to present a couple of figures that show relative parameter importance, and these are the results of multiple model comparisons, and then where you're actually running several different possible multiple regression models and using a model selection tool to pick the best models. And then from among those, you can actually look at how many models contain a certain parameter and how good was the fit or the explanation of variability in those models. And then you, by taking both into account how many models contain something like carbon, which you see on the y-axis here, and how good those models were as far as something like an R² or the lowest amount of error that wasn't explained, we can actually rank the relative importance of these parameters. And we included a number of different things in this model. This is looking at species richness in the upper right. We had age included here, which was actually rejected as something that was significantly impacting species richness in this initial analysis. But carbon was important in every model, as you can see. You can tell that by looking at the lower left-hand corner. And interestingly, biofilters in Sydney, this positive effect of Sydney here, this relative parameter importance weight for Sydney is showing that Sydney tended to have higher species richness than any other city. Brisbane actually tended to be lower than the other two cities as far as species richness in their biofilters. And in the bottom right, abundance, there actually was, we saw an effect of carbon again. It's a very important parameter. Sydney had slightly higher abundance also than the other cities, but as the biofilters got older, there was actually a significant negative effect, or age was an important parameter that actually we saw a decreasing abundance, if anything. This is abundance, number of organisms rather than richness. We saw a decrease in that over time. And we didn't see any strong correlations with individual metals. And part of the reason that might be that, for one, they didn't reach levels, at least compared to toxicity studies. They didn't reach levels that caused significant mortality or reproductive effects in organisms. But like I said, we were analyzing for 26 different metals, and there's some possibility that we might be able to use some sort of a multivariate technique to look at the cocktail of metals and if there was any effect of those on the organisms. And we're starting to work on that now. On slide 20, this is just looking at the different metal for the metals that people are most concerned with as far as stormwater. On the upper left, we have cadmium, on the upper right, copper, lower left, lead, lower right, zinc. Soil depth for all these is on the x-axis, and for each city, which are shown in different colors, Sydney is in black, Melbourne is in gray, Brisbane is in white. For each city, we have soil depth of 2 centimeters, which is including the very uppermost layer where the crust is, and then we have soil depth of 10 centimeters into the soil on the right. So it's 2 and 10 and 2 and 10 and 2 and 10 for each city. And metal concentration in milligrams per kilogram is on the y-axis. What you're seeing here has been shown in laboratory and in other field studies before, that the metal concentrations in the upper layers of the biofilter are substantially higher than they are in the lower layers of filter media. This is really convenient for actually maintenance of biofilters, because these things start to form a crust, which can cause clogging. It's very easy to go in and remove the upper 2 centimeters of soil. That's actually the recommendation now, based on some studies that Belinda Hatt was involved in Melbourne. You can remove the upper 2 centimeters of soil, and you can remove that clogging layer, and in the process, you're removing a lot of those accumulated metals that are in the biofilter. And these differences between these are 95% confidence intervals around these average metal concentrations for each column. And so it is important to notice that there is a lot of variability in here. But the differences there's variability among cities and also within a certain depth. But these differences are statistically significant between the 2 centimeter and 10 centimeter depth. Okay, slide 21. So those are some of the preliminary results. Okay, so yeah, sorry, I should have made a bigger point. We're moving into discussion of metals now. We didn't find any great correlation between metals and carbon content in the upper layer of the soil, but we're actually expanding beyond just looking at internal factors in the biofilters, and we're trying to do a spatial analysis to look at the biofilter age and land use effects on metal accumulation in biofilters. And we're considering factors such as age, as I mentioned before, because it is a chronosequence, carbon content, road surface area, and tree cover. And we're expecting all those but tree cover to have a positive effect, because metals accumulate over time, metals bind to carbon, and with more roads, you have more metals being deposited in the urban environment. And tree cover we're not sure about because it provides carbon and leaf litter, but it also, where there's tree growing, you're obviously having at least some reduction in impervious surface. So we're not sure if that's going to be there'll be a positive or negative correlation with tree cover as far as metal accumulation. Vegetation cover, again, we're evaluating that as well, but we're not sure what the result is going to be. We expect impervious surface to have a positive effect and green space to have a negative effect because it allows more infiltration within the catchment outside of the biofilter. And I want to point out Jimmy Long right now, who is one of the students on the Up-Down-Under course last year. He's at UCSD, and he's currently helping with some of the spatial analysis. We're using Google Earth. Actually, he is using Google Earth right now to take some of these aerial photographs, and he's using Google Earth Pro to actually delineate the surface area that's taken up by each of these different type of land use factors. For example, here you can see he's drawn radii of different color around the point we sampled the biofilter, and within each of those radial distances away from that sampling point, he's actually summarizing how much. And for example, you can see here in blue, he's highlighted all of the road cover. He's calculated road surface area within a certain radius in a certain area around the biofilter. Jimmy's been really great. He's been working really hard on this stuff and also on analysis of some samples from some biofilter core studies. And he's about to graduate. So if you haven't found a good graduate student, I know he's applied several places. I'm not sure if he's accepted anywhere yet. But anyway, looking at some of those results from the spatial analysis, one of the things we found out that was really important for at least 2 metals, and this is on slide 23. Here we're looking at these 2 panels on the right. Melbourne road surface area is in square meters on the x-axis. And these are results, I should say, just from the city of Melbourne to begin with. And we're looking at copper on the upper panel and zinc on the lower panel. And we're seeing a significant correlation between road surface area and both zinc and copper. And looking at a similar plot on there's a positive correlation in the following slide. This is looking at some of the things. We're still actually digitizing some of the images, and so we don't have all the land use factors completed yet. We're about to finish that for Melbourne. And we're also getting GIS surface layers from some of the city councils, but those are slow in coming through, so we should have those shortly. But for now, we're using this other technique to get an idea of some of the trends. But so we have another plot here on slide 24 with parameter importance. These 4 different parameters shown in different colors: road surface area in blue, tree cover in red, age in green, and carbon in some purple. And as you can see, for copper and zinc, you can see that road surface area, as I showed in those previous figures, was an important parameter in predicting the concentrations of those in the surface layers of biofilters. But it wasn't so important for lead and cadmium, which you can see in the middle there. And one of the reasons that it was for this relationship with copper and zinc and not the others is that road tires and brakes actually have fairly high concentrations of copper and zinc, but not very high concentrations of lead and cadmium. So it makes a lot of sense that road surface area will be having a significant effect on this. And actually, Matt Burns is helping with gathering. He's a postdoc working with Tim Fletcher, Darren Boss, and Chris Walsh. He's collecting a lot of the spatial data for Melbourne, and this is one of the things that he had said a while back. He's like, I bet most of this is going to be related to roads. And so far, what we're seeing supports that actually seems to be the case. So slide 25. Now looking at the deeper layers of metals, I'm almost done. On the upper left, these are metals at the 10 centimeter depth. And we have, again, relative parameter importance of a couple different parameters that came through in our final models, among a number of things that we tested. For copper, at best, we're explaining about 57% of the variability in copper concentrations at 10 centimeter depth. And that was being best explained by the age of the system, which had a positive effect. The carbon concentration within the filter media, and that makes sense because copper binds well to organic matter, and also whether or not you're in Sydney. So I know there is some competition between Sydney and Melbourne, and I guess one of the things you can say is that you have lower concentrations of copper in your biofilters in Melbourne than Sydney does. On the lower left-hand corner, we have zinc. Similarly, age was important to predicting zinc concentrations in low depths of the biofilter. The amount of zinc on the surface, as you would expect, was the source that was allowing zinc to sort of trickle down through the porous media into the lower depths of the biofilter. So the upper concentrations matters, lower concentrations are positively correlated. And also the total abundance of invertebrates was positively correlated with zinc concentrations in lower depths, even though it wasn't correlated in the upper layers. And that makes sense because most of these invertebrates, a large number of them are actually worms that tunnel through soils, and they're taking in soil at one place, they're feeding on it, it's passing through their digestive system and being deposited elsewhere. So while in a laboratory you might have all your metals really highly, tightly clustered at the upper layer, if you have things that are feeding on the soil itself and tunneling through it, it makes sense that metal mobility could be increased by certain invertebrates. Similarly, lead is on the upper right corner. There was these models tended to contain more parameters, so we had a large number of things such as age, carbon, whether or not you're in Brisbane. Brisbane tended to have higher lead concentrations. All invertebrates and potworms, they both had significant positive effects on lead concentrations at lower depths, and also the amount of lead on the surface crust. And then cadmium. Cadmium really wasn't related to age like the other 4 metals. It was most strongly predicted by the amount of carbon in the biofilter, and also by cadmium concentrations at the surface. So one last data slide on slide 26. I think it's interesting. I've been thinking lately about, and I haven't heard people talk about it much before, but dealing with non-point source. What I'm showing here is basically one sample from a biofilter in Sydney that was in a fairly industrial area. And there's just a huge suite of metals that are present in one sample, and you can see things like cadmium, arsenic, mercury around this smaller blown out plot on the right. Things that we know have toxic effects in humans and in organisms. And dealing with non-point source pollutants is difficult because oftentimes they're being released in the environment in low concentrations and spread out over a very wide area. One of the interesting things about biofilters is that they're strategically put in locations that filter large catchments from urban areas and are basically sort of taking a record of everything that's been flowing through the stormwater over time. And while a lot of these things like mercury and cadmium may have been in extremely low concentrations in a stormwater flow, like if you collected a sample in a gutter flowing down a street, you might have had cadmium in concentrations that were below detection limits. But you could go to a biofilter, say after 5 or 10 years, and sample the soils, and you suddenly just discover that you have discharges of a lot of these toxic metals within your watershed. And if you know the catchment, you might get an idea of where there might be problems for things that are not necessarily being released in large quantities, but are having long-term releases in the environment and can then accumulate in the receiving ecosystems and have negative health effects for humans or for the organisms present there. So I think biofilters are interesting as potential sentinels and sensors of non-point source pollutants in the environment. And then again, I just want to reiterate that we're just really getting into the bulk of the analysis now, these data sets, and we're doing some more complex spatial analyses and community structure analyses, and looking at this on sort of is on slide 27. I have a picture of Sydney, just sort of thinking about this with a landscape perspective and an ecological theory perspective of island biogeography and also landscape effects on metal accumulation in these biofilters over time. And so just quickly in conclusion, invertebrates rapidly colonize biofilters, and there's unique community structure among sites. Biofilter age doesn't seem to be a great predictor of species richness or abundance. It looks like media characteristics and potentially the surrounding landscape are more important. Similarly, for surface metals, the age was less correlated with concentration than land use was. And also then metals at the metals at 10 centimeters did increase significantly with age, which makes sense because it takes time for them to trickle down through this porous filter media. And in some cases, invertebrate abundance appeared to have a potential effect on metal accumulation in the lower layers. And so now we're working on expanding that analysis, as I just described earlier. I have a lot of people to thank, including NSF, and then tons and tons of field and lab support in Australia, and tons of field and lab support and conceptual and thinking support from collaborators here in California. So with that, I'm probably going way over, but I'll be happy to take any questions if we have time.
Okay, very good. Thanks very much, Andrew. That's a lot of good information. So do we have questions?
Andrew, that was a great talk, and every time I see this, I'm kind of flabbergasted at the kind of numbers that you're talking about in terms of the organisms that are present per square meter, and it kind of made me wonder. I know that your focus at this point has been on heavy metals, and then also just characterizing the community structure in the biofilters, but do you sort of thinking about Brandon's talk and all with nitrogen, looking at nitrogen specifically, and then also thinking back to Perrin's talk yesterday on nitrogen cycling in some of the natural sediments. Do you have a feeling for what the significance of the invertebrates are relative to overall nitrogen pools in biofilters, and then also their function in terms of relative importance of sequestration and/or denitrification?
That's a really good question. Actually, there was that paper that we recently submitted to Ecological Engineering where we sort of Monte Carlo type analysis where we were trying to estimate the total amount of nitrogen in the invertebrates and compare that to the total amount in soils. And it was, even in biofilters, which should have, even in fairly new biofilter, which should have fairly low nitrogen accumulation, they were still a very small percentage of the total nitrogen that was there. And I actually didn't talk about it because I didn't already talked too much today, but we collected nitrogen data on those biofilters as well, and that tends to be correlated with the carbon pretty closely. So a lot of that nitrogen might be locked into the dead organic matter, might be accumulated there, maybe in microbial biomass, probably in or possibly still locked in the leaf litter and things like that, or assimilated into plant roots.
And then function.
So Brandon and I in our column studies, I didn't present any of that today, but we ran a sort of a paired column study together where we had earthworms in some biofilter columns. And we didn't see a huge effect on function in there, and actually we didn't see any measurable effect on effluent concentrations. We also measured greenhouse gas concentrations to see if there were any emissions coming from them. We couldn't really measure any huge differences there. But that was one species of worm, potentially the wrong species. I mean, these things behave really differently. So I'm sure if somebody just redid our experiment and picked something that was, more active or eating more, or doing more in the system, they may have found totally different results. Some of the estimates for the amount of that earthworms contribute to denitrification, you'll see things as high as 60% of the denitrification coming from soils, and from some studies, which I think seems really high, but apparently it's possible. I think those tend to be systems that already have low denitrification. If you look at one of the studies that Eduardo Daly and some of his collaborators at Monash did recently, they were one of the few studies to measure nitrous oxide emissions coming from a biofilter. I mean, they were measuring, at least in the hundreds, of micrograms of nitrous oxide nitrogen per square meter per hour. I think the rates were somewhere around there. I have to look again, but we were measuring somewhere directly coming from earthworms, and the estimations that we did in the paper we submitted recently to ecological engineering, we were estimating more like 2, coming from, compared to something that was in the hundreds, coming directly from the worms. But that's not to say, as I mentioned earlier, denitrification is enhanced in the feces, in materials that they've ingested, because they increase microbial activity. So it's not necessarily what's just coming out of the worm, but also, what's left behind. So yeah, there's potential, but we haven't measured it ourselves yet.
I also wonder if there's kind of a methodological question here too. So we tend to look at biofilter performance in the laboratory by either doing mass balance on the nitrogen that's the soluble nitrogen or the inorganic nitrogen that's in the stormwater, synthetic stormwater, versus what comes out of the biofilter over time, or we label the nitrogen, say N15 labeled nitrate, and then look at its fate. But it strikes me that again, thinking back to Perrin's talk, I'm not going to represent him, but my sense is that that ammonification is a really big factor. If you've got accumulation in these natural biofilters of a lot of leaf litter and organic material, I would imagine that earthworms and the invertebrates in general could really accelerate the breakdown of that material and actually end up releasing a lot of ammonium, which could then essentially result in leaching of ammonium, or if it's nitrified nitrate. So, anyway, I maybe get you to comment on that, and then sort of I'll also reflect on the sort of the nature of the experiments that you were doing and whether we would gain insight into those kinds of issues.
No, I mean, that's a really good question. And I mean, a lot of studies in natural systems have found that it seems pretty common for a lot of invertebrates to increase the downward movement into soils of ammonium and nitrate and things like that, and organic nitrogen. So they're actually getting those things to leach down into the biofilter, and where they're probably more likely to have an effect on things like denitrification. I'm not sure if increasing denitrification or ammonification, or both potentially, if they're cycling together in that way, is a positive, because they're bringing potentially organic matter from the surface down into the system. It could be a negative effect. We just don't have enough data yet to say whether it's a good or a bad thing.
I'm also wondering if it's something that would be interesting to model, basically looking at that, assuming various functional roles for the inverts, more vertical migration, breakdown rates of organic material, and so forth, and just sort of play some scenarios out to see, what role they could play in terms of nitrogen cycling in these systems.
Definitely, yeah. I mean, well, we have this. I think this data set that we've collected is really ready for that kind of work, and we're happy to share it, because I think that this could be used for a lot of different things like that. It's unfortunate that we didn't, go out and collect these abundances and then measure rates or fluxes of things at the same time, because things may have changed since we were actually collecting these samples. But it's for example, that email that you received from Paul Tatham in Sydney. He was going around measuring infiltration rates in the Sydney biofilters. I'd love to see if that matches up at all with the invertebrate abundance that we found in these places. But again, it's a year or two later, So, but yeah, I think these data certainly are welcome to be used for that kind of thing. And it'd be great to do a repeat sampling where you actually measure fluxes when you're out there in the field.
Thank you, Andrew. Great talk.
Thanks.
So I have a question about your order versus age, or order richness versus age. So do you think that you might get a pattern if you didn't throw all the orders together, but you just look? Because it could be that, you know, there are some taxa that come in with the plants, so they're always going to be there, but there may be others that aren't, that then they might have a relationship with age in that case.
Yeah, no, I mean, that's what we're hoping to do a little bit more of. And that's a really good point. Like, you know, there might be certain ones that are really mobile and come in really early, and you know, you might see patterns of succession over time. Just looking at orders is kind of it's almost like a black box, right?
Yeah.
There's a lot more, and we can also, we're working on actually identifying them down a little bit farther to get as much data as we can. It's time consuming, as you know, but yeah, we're trying to get a little more data out of it that way. And you've also mentioned, and you've made the comment to me in the past where you thought it'd be great if we could go and sample this, the soil around, you know, the nearby soil and the lawns nearby and stuff, and see what was there, because that would have, I mean, that would have been, oh, that would have been great. But it takes so long already, you know, to look for the source populations, but it takes so long to go through these cores as it is, you know, to add more onto what we already had. It was over 120 soil cores, I think, that, you know, we're looking for tiny things, and so it takes a long time to go through it. What you're talking about would be a dream data set, but we
Right.
You know, yeah, so yeah, but hopefully we'll be able to get at some of those things you're talking about a little bit with the data set that we have, tease apart some of those early colonizers and things that disappear as they get older. It was interesting because, sorry, I just was thinking about how Brandon was talking about some things you guys have seen with your plants and the diversity over time, you know, in some of the older systems where it maybe doesn't seem quite as high. And, you know, it's possible in some of these, we're seeing something similar with invertebrates too, which would be really interesting if those things were comparable.
Yeah, which makes me ask you a totally off the wall question, because you're looking at the inverts in the soil. I know you're not looking at the inverts that are above the soil, but do you have any feel for what the link is, if there is a link between what's above ground and below ground, and anything like that?
I think based on what you see in the literature, there should be. I don't have any data at all, unfortunately, to be able to test that myself. But I mean, you know, if the ones below ground, a lot of times what's happening above ground is feeding the below ground, you know, with reed exudates and things. And if what's happening below ground is increasing plant growth and nutrient uptake, it should be benefiting the herbivores and things, you know, predators that are feeding on them above ground too. So yeah, it should be. I mean, there should be linkages. But yeah,
Yeah, so
unfortunately.
that's an even more dream data set.
Yeah.
Right? Because not only is it around, but it's also looking below and above.
Yeah, we need to hire, you know, half a dozen people and redo this. I think we could do it really great. Yeah.
Yeah. Next grant.
Yeah.
Yeah, I actually.
Any other questions?
Yeah, I do.
Okay.
Sort of based on what actually I asked Lisa earlier about age and how you determine sort of the age of these systems, given people come in and maintain them and do a variety of other things to them. So I was just wondering, in terms of some of your associations, sort of relationship with age, some of them didn't. If we know that as new plants come in, some of those invertebrates come with them. Sort of, can you speak on sort of what your thoughts are relative to how we classify age of these systems and what that means for them?
Right. No, that's a really good question. You know, there's a huge biofilter here next to Scripps, where that they're getting ready to basically replace all the filter media. You know, take everything out and put new filter media in and redo it. And I consider that, you know, like it's like being born again, right? I mean, that's starting over at zero. Even though the surrounding source environments are still there and providing, you know, feeding with invertebrates, but when you're talking about maintenance, we're scraping off the surface layers to get rid of the crust and the metals and stuff. I mean, that's not really resetting the system entirely, although it's a disturbance. But so that's one of the things we're doing, actually, because, you know, we're thinking a lot about that. We're trying to get any maintenance records from the city councils of, you know, have they gone out and replaced, you know, what was there, which would you know, change our whole relationship with time, you know, I think. So yeah, that was a really good question.
Yeah, it's something I've been wondering myself when I've been looking through sort of the studies that have been looking at fecal indicator bacteria removal, when they say it's like, well, the system is X number of years old, but we replanted it before we did the study. It's sort of a, what does that mean in terms of how old it is?
Yeah. Does the age really mean anything anymore?
Yeah. At that point, yeah.
Yeah. Yeah. No, yeah. It could affect all kinds of things.
Mm-hmm.
So Andrew, just a follow-up question on the modeling of worms. I just want to write a paper that has modeling of worms in the title. Do do you think you have scenarios that would be feasible to test? Like, for example, migration patterns, or, I mean, are they like birds? Do we know, do we know when they fly south, or are they more just a random, diffusion, sort of a biologically enhanced diffusion process?
Yeah, no, I mean, it depends on which ones you're talking about. Earthworms tend to move a lot more during wetter conditions, and some people thought, oh, they must be coming out because they're getting flooded and they can't breed or something. But a lot of times it's just, because it's easier to get in and out of the soil when it's raining, because things are wet and it's easier to penetrate soil layers and explore. And you don't run the risk of desiccation because they're getting oxygen they're actually getting oxygen directly through their cuticle. So, I mean, earthworms tend to move when it rains, but, other things, if it's something that actually has fully metamorphoses, I guess it's going to depend on its specific life cycle. The species of worm probably matters, and they also go and follow their prey. So one of the things that we've seen that's interesting is that we have certain spiders that just show up in biofilters that have isopods because they specialize on that group. And earthworms might, okay, now I'm going off on a tangent, but you just want to know about worms, right?
Whatever you want to talk about, man.
No, I think that with worms, I mean, there's a number of different things that could affect it, and I don't really know. But it probably depends on which group of worms that you're looking at, and you could model that. You could run different scenarios with different worm communities, and it could get really complex.
Thank you.